Remember, we are trying to make ATP energy. Let's take a look at this formula. Remember that reactants, the inputs, are on the left side of the arrow, and products, the outputs, are on the right side of the arrow.
What is cellular respiration? It is the releasing of energy that can be used to do the work of a cell, and it is completed in 4 steps. Step 1, glycolysis. Step 2, acetyl-CoA synthesis. Step 3, citric acid cycle. And 4, the oxidative phosphorylation or the electron transport change. In step 1, it is glycolysis, which fuels molecules are partially broken down, producing the ATP and electron carriers. This occurs inside the cytoplasm. Glycolysis is the production of pyruvate by creating 2 ATP molecules and using NADH. The NADH can be used to create 5 ATP molecules, so the total amount of ATP molecules produced in glycolysis is 7. Throughout glycolysis, there are 3 phases. Phase 1 is the consumption of 2 ATP molecules creating ADP. Then phase 2 is the cleavage point of the cycle. This is the separating to create 2 separate pyruvate molecules. In phase 3, it is the production of 4 ATP molecules from ADP, adding an electron onto itself, and of 2 NADH molecules. Stage 2 of cellular respiration is the formation of acetyl-CoA. Acetyl-CoA is the first substrate in the citric acid cycle. This is part of the cellular respiration, also occurs in the cytoplasm. Formation of acetyl-CoA occurs inside the mitochondrial matrix, which is a space inside of the interior membrane. The first part of the formation, pyruvate, is oxidized and then split off to form carbon dioxide. The electrons lost in NAD+ is reduced to NADH. The remaining part left from pyruvate, called the acetyl group, or CH3OC negative, is now transferred to coenzyme A. After being transformed to acetyl-CoA, it is then carried to the next set of reactions. During the formation of acetyl-CoA, there was no ATP formed, but 2 NADH molecules were, which creates 5 ATP. Stage 3 of cellular respiration is the citric acid cycle, which fuels molecules are broken down, which produce ATP and electron carriers. It takes place in the mitochondria. The citric acid cycle is the transfer of hydrogen protons and electrons to NADH. The citric acid cycle is also called a cycle because the starting molecule oxalate is regenerated at the end of the cycle. There is a release of CO2 throughout the cycle. This is where carbon dioxide comes from what we exhale. Energy that is released in the cycle is then transferred to NADH. 3 molecules of NADH and 1 molecule of FADH2 per turn on the cycle are produced. During the cycle, 2 ATP molecules are produced along with cNADH molecules, which create 15 ATP molecules. Then, 2 FADH2 molecules are produced, which can create 3 ATP molecules. Therefore, 20 molecules of ATP can produce in the citric acid cycle. Stage 4 is the oxidative phosphorylation, also called the electron transport change. And this is the last stage of cellular respiration, and it occurs in the mitochondria. Electrons are transported along a series of 4 large protein complexes that form the electron transport chain. Complexes 1 and 4. Electrons donated from NADH are complex 1, and electrons donated from FADH2 enter through complex 2. Then they were transferred to complex 3 and complex 4. When oxygen accepts the electron, it is then reduced to water. ATP, H2O, and H+ is produced in the part of the cellular respiration. ATP synthase is the last piece on the electron transport chain. This molecular machine is composed of 2 different subunits, F0 and F1. F0 is a membrane-spanning subcomplex, while F1 is a soluble component. F0 forms a channel in the inner mitochondrial membrane through which protons would flow through. F1 is the unit synthesizes ATP. Phosphofructokinase 1 stands for PFK1. It is a regulation of PFK1 can determine how fast and slow glycolysis and the rate of ATP production. This figure shows a reaction with activation and inhibition. Activation is where the ATP levels are low. The PFK1 will be activated to continue glycolysis. This is showing that ATP and AMP are being inputted. With inhibition, when ATP or the citric levels are high, the PFK1 is inhibited, causing glycolysis to be slowed down. And this shows that ATP and citrate are being inputted.